share my screen so you can see everything. What you've got there right now is your virtual class. I guess everybody is familiar with that. And you've got here this unit that is the beginning of biology, which is the unit that we were working with these days. Cells, the building block of life. With this group, with your group, we are doing quite well. So we've seen a lot of things. We've seen these 5 sections, which is what we've got in Unit 1 in your book. Remember that this exam was based on 2 units in your book, but it's just half of Unit 1 and also half of Unit 2. So these 5 sections, prokaryotic cells versus eukaryotic cells, the comparison and the evolution is all in Unit 1, apart from the evolution. Remember that evolution was represented by this picture, the evolution from prokaryotic to eukaryotic cells, the endosymbiosis theory that Lynn Margulis proposed. This picture, you've got it in, I think it was page number 80 in your book. And is also part of this exam. Okay, it's just this part. Remember this, well, I'll go back to this in the review. Then what is the nucleus of the cell? The parts and the function of each part, the coordination between the nucleus, the rough endoplasmic reticulum, Golgi body, and ribosomes, the function of the centrosomes, and how do cells divide and why they divide. All that is included in Unit 1, and we already saw all those sections before Christmas. We also started with this part, that is the last part of the unit, and it will also be included in the exam. Alguno pensaba que empezábamos a 1:30, no sabéis que por el COVID tenemos los recreos estos cambiados. Chromosomes and cell division, it's information that you've got in Unit 2 in your book, and but it will also be included in this unit for this following exam. So apart from the information in your book, we've been going through this PowerPoint presentation that you've got there, cells, the building block of life, which is this one, in which we saw the definition of a cell as the smallest unit of life, the functional and the structural unit, components of the cell. We also saw the following part, that is prokaryotic cells versus eukaryotic cells. So that prokaryotic versus eukaryotic. And you've got here the characteristics of prokaryotic, which is essentially what differentiates prokaryotics from eukaryotic. So because I want you to participate a little bit, I will go one by one. Mañana ya no se me conecta ninguno después de haber ido one by one today. Sí. So Aitana, you're the first one in my list. Can you name one difference, either with your microphone or writing on the chat, one difference between prokaryotic and eukaryotic cells? Quiere Lucía ser la primera y contestar ella. Aitana, are you going to use the chat or your microphone? Most important, perfect. Most important difference between prokaryotic and eukaryotic cells.
Eukaryotic cells have nucleus and prokaryotic cells don't.
Okay, so that's the first one, the most important difference. But we also know 2 more things. What about the size? Which one is bigger?
Eukaryotic ones.
Okay, eukaryotic cells are bigger than prokaryotic cells. In fact, we said that the size of this cell is more or less the size of this organelle. Okay, so that's another difference. And the third one is the kingdom in which we find each one. Prokaryotic cells are only found in one kingdom. That's why these organisms belong to a different domain. Eso lo veréis el año que viene, lo de los dominios. What's the only type of organism with prokaryotic cells? Starting with B, Aitana, for you, the only type.
I don't know, teacher.
You don't know? Anyone that wants to help here? Maybe writing on the chat or just opening the microphone. Miguel, what's the only type of bacteria? Perfect. Bacteria is the only type of organism. They belong to the Monera kingdom. So that's what you've got here in this slide. Prokaryotic cells, they lack of a nucleus. They don't have a nucleus. The genetic material is dispersed in the cytoplasm. It could be in two different regions, the nucleoid or the plasmid. Remember, most of the DNA is here in the nucleoid, but scientists have discovered that some bacteria, not all of them, some bacteria have this plasmid, maybe 1, 2, even 3 plasmids that have this circular shape, and they contain some DNA. The scientists related especially with resistant bacteria, those that are difficult to be killed with antibiotics. Well, the only thing you must know is that the DNA could either be in the nucleoid or in plasmid. Then the second difference is that they are present only in the Monera kingdom, only in bacteria, and the size. They are 10 times smaller than eukaryotic cells. Okay, so those are the 3 differences that summarize this part, comparison. And then on evolution, we've got, summarizing this evolution, we've got this very important diagram proposed by the female scientist Lynn Margulis. Well, I'm going to ask someone else, next one in the list, which is next one in the list, which is Alvaro. Alvaro, either using the microphone or writing on the chat. I want you to tell me, looking at this picture, more or less what the picture represents, or why. What are the evidences that Lynn Margulis used? Try to tell me, try to explain something about this diagram. Míralo así despacito. And we've got on the left, what do we have on the left? Which type of cell is on the left of the diagram? No sabes, profe, hombre, está aquí. Which type of cell? ¿No ves mi cámara? Mi pantalla?
Ah, sí, sí.
Which type of cell is on the left?
Pues...
Is it eukaryotic or prokaryotic? Okay, we've got a prokaryotic cell, the original prokaryotic cell. So we're talking about something that happened hundreds of millions of years ago. ¿Vale? This is a process that probably happened hundreds of millions of years ago. One original prokaryotic cell, because hundreds of millions of years ago only prokaryotic cells existed in the planet, one of these cells suffered one process. What's the difference that you see between this picture and that picture? ¿Qué vemos? What's the difference? This label there. Perfecto, ¿verdad? And what does it mean? Enfolding means that the plasma membrane, we don't know why, probably by some mistakes, because that's how evolution usually happens, due to mistakes. probably due to mistakes, the plasma membrane of this prokaryotic cell enfold a little bit. Sometimes when mistakes happen in nature, it means that the organism will not survive because a dangerous mistake has happened. Imagine that you are born with a mistake in your heart. That probably means that you are going to die very soon because your heart doesn't work. But sometimes the mistakes led to one structure that is better prepared for the environment. This is what happened, probably happened to one of these bacteria, that the membrane enfolded, and that was a good thing because those enfolds protected what, Álvaro? This membrane was protecting what?
The DNA.
The DNA. So we still don't have a nucleus. Esta todavía no es eucariota. We still don't have a nucleus, but we've got a DNA that is a little bit more protected. So these type of cells will be better prepared to survive in the environment. Then they have descendants and more descendants. So this didn't happen in one day, que a veces pensáis que esto es un día solo. This is generation after generation. Those cells started having more and more enfoldings, and then some of them developed this structure, the endoplasmic reticulum, the Golgi body, the nucleus, all those membranous structure. And we can already say that we've got a eukaryotic cell. This one has a nuclear envelope, so this could be the first eukaryotic cell with a nucleus that is protecting the DNA. That is good because this information is important, that is protected. Then, why did Lynn Margulis developed this theory? It's just that she had a happy idea. Well, she thought about that because of something that we've already seen. We've already seen that the eukaryotic cells have this nucleus, and the nucleus has a double membrane. And if you see the endoplasmic reticulum, that also seems to be kind of a double membrane. So it could be the result of this membrane enfolding and then forming this structure. Okay, so important thing that we must know about this endosymbiosis, the way it worked. Millions of years of evolution led to the enfold of this plasma membrane that, because it was a good thing, it was maintained, forming the first eukaryotic cells. But then the endosymbiotic theory has these two branches, one forming the first heterotrophic eukaryotic cell with mitochondria, and the other one forming eukaryotic cells. Vamos con el siguiente, dejamos descansar ya a Álvaro y tenemos a Amalia. Amalia, could you please turn on your microphone or write on the chat what you consider is the answer for this question? What is happening in this picture represented there? You've got an important label there. Amalia, are you ready? Si alguno sabe que es que ella no tiene micrófono o que no puede escribir ninguna de las dos cosas, que me lo diga. Si no,
sí que tengo el micrófono, pero...
Venga, pero no sabes contestar.
Exacto.
Anda que no, mira el cartel. What is going on there? Can you read these letters or you don't? Más grande todavía. What is saying there? A difficult term, that is engulfing. What does it mean? ¿Qué será engulf? Engolfando parece que no es. What's the translation for engulf? Any volunteer? Someone that wants to help here? What is engulf? Mira que no te ayudan. Miguel dice que sí. Engulf is?
Engullir.
Engullir, ¿vale? So, engulfing of one heterotrophic prokaryotic cell. What's going on here? This cell that was advanced because it contained a nucleus engulfed a tiny prokaryotic cell. But when it engulfed it, instead of destroying it, instead of digesting it, this cell, by any reason, decided to do a symbiosis. What is a symbiosis? I give you something, you give me something. What is the big cell giving to the small cell? A place to live, a safe place to live. What is the small one going to give to the big one? Energy. It's going to be able to produce energy. So from that moment, this cell already had structures, helpful bacteria, that were going to give them one help. They were going to give them energy. Is this cell better prepared now? Pregunta fácil, que solo es yes or no, que es así que te la sabes, Amalia. Is this cell better prepared now?
Yes.
Yes. so it's going to be the chosen one is going to be selected by nature and is going to go on evolution because it already has other cells that are going to help her producing energy. So it's going to be easier for her to produce energy. Okay, so this one is going to evolve and I already have a eukaryotic cell. Then the following question, the difficult one, in case anyone remembers that. What is the reason that led Lynn Margulis think that this could be the path that this original eukaryotic cell followed before having mitochondria inside? What is the clue? ¿Qué le ayudó a ella to think that this happened in evolution? A ver quién sabe. There's two evidences. No es que tuviera una idea feliz. There's two evidences. Miguel knows it and is going to give us one.
Because both chloroplasts and mitochondria have their own DNA inside them.
Perfect. That's the most important evidence, that when scientists looked at mitochondria and chloroplast, they realized that they contain some DNA and it's not exactly the same DNA as we've got in the nucleus. So it probably comes from a different origin, a different ancestral cell. So that could be the most important evidence. The second one is the size, because remember I told you that this prokaryotic cell is more or less the size of this mitochondria, or in the case of plant cells, the chloroplast. So Lynn Margulis developed this theory saying, first of all, enfoldings, more enfoldings, then engulfing one organism that was able to produce energy, and it turned up being mitochondria inside the cell. And this other branch is explaining the origin of what? What is it explained? Anna, your turn. What is explained in the second branch here? The origin of what? Te dejo el cartel para que lo veas bien. Ana, the origin of... A ver qué ha escrito, a ver si es que sí, qué es más difícil así. Dice por aquí, no tengo micrófono, pues escríbemelo por aquí. The origin of what? What does it appear there? Origin of Plastid, well, plastid is this, which means an organelle like chloroplast, similar to chloroplast, but is the one in bold, es en negrita. The origin of photosynthetic eukaryotic cells. Of course, there are photosynthetics thanks to these plastids. Okay, so that could be one answer. But the answer that I was looking for is the origin of the first photosynthetic eukaryotic cells. What is the origin that she proposed? Something similar. This original cell that was already able to do cellular respiration engulfed one prokaryotic cell that was able to do photosynthesis. Because prokaryotic cells that were able to do photosynthesis appeared before eukaryotic cells that were able to do photosynthesis did. So the order would be more or less prokaryotic cells not able to do photosynthesis. They were chemiosynthetic, then photosynthetic, then eukaryotic, and then eukaryotic photosynthetic. Eso es más o menos el orden. So that's the explanation that Lynn Margulis did in order to explain this origin of photosynthetic eukaryotic cell. So having that, we already have the comparison between prokaryotic and eukaryotic cell, 3 differences, and the process of evolution explained by Lynn Margulis. Next thing you've got here is the nucleus of the cell and the functions. So what you must know about the nucleus of the cell and the function is what you've got in the following slides. First of all, the parts of the nucleus: nuclear membrane, nucleoplasm, nucleolus and chromatin, and the function that is containing the genetic material. So following question that goes to Andrés. Andrés, which component do you prefer? Nuclear membrane, nucleoplasm, nucleolus, or chromatin? Which one do you want to explain to your classmates?
Well,
Venga.
nucleomatin.
Nucleomatin no tenemos. Tenemos nuclear membrane, nucleoplasm, nucleolus, or chromatin.
Ah, vale. Nucleolus.
Nucleolus. Okay, so you're going to explain which part in this picture. Where is the nucleolus there?
In the more pink zone.
This
no,
part.
right.
Esto es el nucleolus.
Yeah.
Is it what you're going to explain? ¿Eso es lo que quieres? Or do you want to explain the pink part?
I don't care.
You don't care?
One.
The purple one. Okay, this one in the middle. Okay.
Yeah.
Nucleolus. They are one structure that is found inside the nucleus. When there's only one, we say nucleolus. If there's more than one, plural is nucleoli. Okay, in case you find a different term. Nucleolus, nucleoli. Do you know the function of these parts of the nucleus?
Maybe protect the DNA.
No. Okay, if you want to talk about a structure that is protecting the DNA, then you're referring to the pink part. Parece que te quieres ir a la rosa, así que vamos a explicar la rosa. Venga, the pink part. What's the name of the pink part?
Nucleus.
Nuclear.
Ah, vale, the nuclear envelope.
The nuclear envelope, nuclear membrane. Okay, the nucleus is all that. Vale, estamos aquí. That's the cell. Inside the cell, we've got the nucleus, and what is represented in this picture is just the purple part. Okay, so this part. Nuclear membrane in pink. What would you say about the nuclear membrane? If I ask you something about the nuclear membrane, you've got to mention 2 things. First of all, this, and the second part is this. Venga, ¿qué me tienes que decir? Esto. It's a
membrane.
It's a membrane. Is it simple or double?
Double.
It's double membrane, 1 and 2. Remember, that is one of the evidences that Lynn Margulis used in order to explain the infolding of the membrane. So it's double, and it's important that it's double. And what about these yellow things? ¿Eso qué son? It's double and it contains... Mira que voy para allá.
Nuclear pores.
Nuclear pores. Okay. So the function, as you said, is protecting...
Well, not the DNA, but the...
Yes, the DNA. Protecting
Well,
the DNA. Perfect. Protecting all the genetic material, DNA and RNA. But having this mission, protecting this DNA, imagine you've got a security box, una caja fuerte. Would you build your security box with holes?
no. It's
Not
better.
really.
Yeah.
It's better if it hasn't these holes. Then why is it important that this membrane that is protecting the genetic material has these holes?
To let the oxygen come in or not?
No, I don't need any oxygen inside. Only mitochondria need oxygen, and mitochondria are located outside the nucleus, so no oxygen is needed inside. In fact, nothing is going to get inside. If the pores are not there to let things get in, what is their mission? Si no están para que entren cosas, what is their mission?
To let things left the...
To let things go out.
Yeah.
They are not going to let anything get in, ¿vale? Nada pasa, de hecho están diseñados para que nada pase, but some material is going to go out. Primera cosa importante, what is the material that is going to go out? Is DNA ever going to go out of the nucleus?
No.
No. Entonces, si no sale, DNA...
The copy of it or...
The copy, that is the RNA. The copy of the DNA, which is the RNA, which is smaller, is going to go out. Esto es lo que decía este dibujo de aquí. Os lo pongo grande para que lo veáis. DNA is a long molecule and it's also thick. It's so thick and so big that it's impossible for DNA to go out through nuclear pores. But this big molecule is going to produce tiny copies, fragments that are going to copy just one part of this long molecule. From the long molecule of DNA, they just copy a tiny part, and they form messenger. This M stands for messenger RNA. ¿Por qué le llamamos messenger? Because it sends messages, ¿vale? Es el que manda mensajes. Sends messages from the nucleus. Is messenger RNA. And it's small enough, it's just one thread, solo una hebra, and it's short, small enough to go out through the nuclear pores. Apart from the RNA, the only thing that is going to go out of the nucleus is ribosomes, los ribosomas, that are produced in the nucleus and need to go out to the cytoplasm. Bueno, pues con esto os he contado, bueno, no os habéis contado, así, two parts. The nuclear membrane that contains this, it's double and it contains nuclear pores. And we've talked about the nucleolus, but very little. Vamos con el siguiente, ya puede usted terminar y respirar. Andrés, Candela, it's your turn. Vamos allá. Which one do you want to talk about? Nucleoplasm, nucleolus, or chromatin? What do you prefer? Candela.
The nucleoplasm.
The nucleoplasm. Venga, si me toca algo, pues esto. That's an easy one because the only thing you've got to say is...
that is a jelly-like substance that is a liquid in the nucleus.
It's the liquid in the nucleus. In the same way that we called cytoplasm to the liquid in the cell, nucleoplasm is the liquid in the nucleus. And what is the only mission? Well, just to let the DNA float in the nucleus. If it didn't have this liquid, the DNA would be attached to the nucleus and it wouldn't work. Okay, so it's just maintaining the DNA, let the DNA move, produce the copies. So it's going to help the DNA working. It's important, but the only thing we've got to say is that it's a jelly-like substance. So we've got the nucleolus and the chromatin that go for the next one. Celia, nucleolus and chromatin go for you. Which one do you prefer? The nucleolus. Okay, the nucleolus, as Alvaro said, is this circular structure in the middle. We could have one or more than one. It is mainly made of which molecule? Is one nucleic acid? Teníamos dos. Which one do you think that is the most important component in the nucleolus? One nucleic acid. Ánimo. It could either be DNA or RNA. ¿Cuál prefieres? RNA, perfect. The difference between the nucleolus and the rest of the chromatin is this rounded structure, the nucleolus, is made of RNA, ribonucleic acid, while chromatin is mainly DNA. And it has an important function. It's going to build one organelle. A ver si has estado atenta. What is the organelle that is built in the nucleolus? The only organelle that is produced inside the nucleus. ¿Cuál será? ¿Alguien ayuda a Celia? What is the name of the organelle that is produced in the nucleolus? Miguel nos levanta la mano para decir.
Ribosomes.
Ribosomes, okay. Ribosomes in this picture are represented by those tiny red dots. Si os fijáis, what are the other organelles? Mitochondria. Is it possible for mitochondria to be produced in the nucleus and go out through the nuclear pores? ¿Caben por ahí? No way. Mitochondria, centrioles, smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi body, even vesicles or lysosomes, none of them are small enough to be produced in the nucleus. The only organelle that is produced in the nucleus is these tiny dots, esos pequeñitos, that are ribosomes. The mission of ribosomes, remember that you've got it in your book in page number 14. And you also have there the structure that you can see with electronic microscope, that is these two subunits, okay? What the ribosome is doing there in your picture in page number 14 is reading a thread of RNA. That is a chain of RNA, messenger RNA, that is being read by the ribosome. So we already have nuclear membrane, nucleoplasm, nucleolus, and the last one is chromatin. Chromatin is made of DNA and proteins, so the mission of this chromatin is it contains the genetic information, the genetic material that has all the information for the cell to work. Okay, so we got here the mission of the nuclear membrane. Next one, the nucleolus. It says here ribosome assemble. It means that it joins all the two pieces, so you form it, forms ribosomes. And in chromatin, we've got this structure. Well, we've got there the first question for the exam. This is going to be one question in the exam. Lara, for you, what's the difference between these two terms? Chromatin here and this one there.
I don't know, that the chromosomes are formed in the chromatin?
Are formed by chromatin, so using chromatin. And what is the difference? That here chromatin is, ¿cuál es el verbo?
I don't know.
Ahí, míralo, no veo mi pantalla.
Condensed.
Is condensed, okay? Is coiled, que significa espiralizar, coiled. Is coiled. So this that you've got there is the DNA molecule, and it starts coiling around proteins, and it coils and coils until it forms chromosomes. When will it happen? ¿Cuándo pasa? Only. Sigue leyendo, no te pongas nerviosa y lee solo eso.
When the cell divides.
Okay, only when the cell is going to divide. So if you look at a cell, if you take one cell and you look in the microscope, if it's a good microscope, you're going to see the nucleus with a material that you're not going to identify. But sometimes in specific cells, you can see this structure. If you see it, means that the cell was dividing. So if you want to find cells with chromosome, the best place in which you can look at is parts of the organism that were growing. For example, we usually do it with roots, con las raíces, with roots in plants. We let them, for example, you take an onion and you let the roots grow in, and then you cut part of this root, and you're going to find that some cells were dividing, and then you see these chromosomes. Each molecule of chromatin is going to form one chromosome. So the last question, la última, que no es difícil para ti, Lara. How many chromosomes would you see in a human cell? How many chromosomes do we have? Venga, un número así, un número bonito. Dime un número, yo ya te corrijo y te digo si sí o si no.
2 billion, I don't know.
2 billion, no, eso es demasiado. In each cell, en cada una, tienes el mismo número en todas. How many? Venga, un voluntario por ahí. Bueno, no voy a coger voluntario, voy a coger el siguiente. ¿Qué me dicen? Por aquí tengo dos manos arriba, sí, Rodrigo, venga, dime. Rodrigo, que tú no me has dicho nada. How many? 7? No, more than 7. Venga, que vamos a hacer aquí un bingo. Laura, ¿cuál es tu bingo?
27.
27, more than 27. Lucía, more than 27.
30.
30. Bueno, ese número me gusta más, que no es, pero por lo menos, at least. In humans, you've got to give a pair number, because half of the chromosomes come from your mom, half of the chromosomes come from your dad. So is it possible that we've got 7 or 47? No way. 30, it could be, but it's not the right answer. The other, Lucía. Venga, un número par, ya tengo muchas manos arriba.
34 or 36.
34 or 36, I'm not sure. Bueno, Macarena, dales una idea, anda.
46.
46 chromosomes, perfect. In humans, we've got 46 chromosomes, at least those humans that do not have any chromosomal abnormality, because we know that people suffering Down syndrome, they've got 47 instead of 46, okay? So in every cell in your body, you're going to have 46 molecules of DNA forming your chromatin. And when those cells are going to divide, you would be able to see 46 chromosomes, each one with these two parts. We already saw the names of these parts. ¿Os acordáis que habíamos visto esto, que era un poco de lío tantos nombres? Decíamos, in a chromosome, the last part, the external region, is the telomere, which was important to protect the rest of the DNA. These telomeres were damaged as you get older. So old people have less size of these telomeres. Every division you lose a little bit. This external part is the telomere. The central part is the centromere. Y decíamos, one question could be, what's the difference between chromatin and chromosome? La segunda podía ser between chromosome and centromere, y hablando de palabras parecidas, the centromere and this organelle here, that is the centrosome formed by centrioles. Teníamos todos esos nombres parecidos que hay que ir diferenciando, ¿vale? So, chromosome, different from chromatin, and it has these regions, telomere, centromere, arm, which is just this, or chromatid, if it's all this, ¿vale? One chromatid had 2 arms, and we said that the shorter arm was always the P arm, and that's the Q arm. Okay, so different structures that we must know from this chromatin. Well, the following part in your unit, the cell's nucleus and its function, the next one is the coordination between the nucleus, rough endoplasmic reticulum, Golgi body, and ribosomes. So if you see in the presentation, we've got the next thing, which is the rough endoplasmic reticulum. There's 2 types of endoplasmic reticulum, but this year, the smooth one, we are not going to see anything. We are going to focus on the rough endoplasmic reticulum that contains ribosomes. Because the mission of ribosomes is synthesize proteins, they are going to build proteins. The rough endoplasmic reticulum that contains these ribosomes is going to have something to do with proteins. So, next one, por aquí. Cintia, ¿tienes la mano levantada porque te has despistado o quieres decir algo?
Es que no sé bajarla, profe.
Pues le vuelves a dar otra vez. Aquí en la barra esta, si pulsas la levantas y si le vuelves a dar otra vez, la bajas. ¿Tienes esa barra tú?
Sí, ya, sí. No.
Bueno, es igual. Sabiendo que no quieres nada, si quieres algo, abres el micrófono y ya está. Pedro, que te veo por aquí. Pedro, could you please explain what's going on in this picture? What is this red structure representing?
A ribosome.
A ribosome. We have this red ribosome with 2 subunits, similar to the one that you've got in your book. And is reading, what is this ribosome reading?
RNA.
RNA, but it has an M. ¿Te acuerdas que he dicho yo que era la M?
No.
Messenger RNA, because it's sending messages. Okay, so this messenger RNA that comes from, ¿de dónde viene? ¿Dónde había sido producido? Sí, hombre. This messenger RNA was produced where? In the nucleus, using DNA. Eso es, ibas a decir eso, ¿que sí? Using DNA, the copy is the messenger RNA that goes out of the nucleus through the nuclear pore. So, this messenger RNA comes from the, ahora te dejo decirlo, comes from the nucleus, and it gets into the ribosome, and the ribosome has an important mission, that is, read the messenger RNA in order to produce, ¿qué forma? The ribosome. Proteins. So this purple molecule is a protein that has been produced in the ribosome thanks to the information given by the RNA, the messenger RNA. I already have a protein. If this ribosome was in the cytoplasm, que teníamos algunos, ¿verdad? There are ribosomes dispersed in the cytoplasm. If the ribosome is in the cytoplasm, it's going to produce a protein, and this protein will be directly used in the cytoplasm, and it's going to be a simple protein. Estas son simples, ¿vale? But if the ribosome is not in the cytoplasm, but is like those ribosomes in the, ¿cómo se llama esto morado?
Lo morado.
Sí.
De nuclear membrane.
Que estoy señalando.
Ah.
Bueno, azul oscuro, añil, no el morado. Es esto.
The rough endoplasmic
The
reticulum.
rough endoplasmic reticulum. If the ribosomes are in the rough endoplasmic reticulum, they will do the same. They will produce proteins, but the protein will not go to the cytoplasm, but the protein will get inside the rough endoplasmic reticulum, which is what you see here. What you see here is the inside of the rough endoplasmic reticulum. Why? Because this protein is going to turn into a complex protein. So what is the mission of the rough endoplasmic reticulum? Receives the protein, makes them more complex, and stores them, las almacena, stores them. Then after storing them for a long time, or a short time, depends, it's going to send them in these bags. How do we call these bags, Pedro? ¿Lo ves ahí?
Protein inside transport vesicle.
Okay, so they are vesicles, transport vesicles, because they are going to be transported somewhere else. Where to? From the rough endoplasmic reticulum to?
To the Golgi apparatus.
Perfect, to the Golgi body or Golgi apparatus. ¿Veis aquí que dice coordination between the nucleus producing messenger RNA, the rough endoplasmic reticulum with ribosomes that receive this messenger RNA and produce proteins, and then the rough endoplasmic reticulum gives them to Golgi body. Eso es lo que tenéis en este punto, que sería este de aquí. So this endoplasmic reticulum produce this complex protein and send it to the Golgi apparatus, Golgi body. Okay, so next one is for Raúl. Raúl, Golgi body receives this vesicle with the protein. What is it going to do? Te dejo la pantallita para que no te quejes. ¿Tenemos a Raúl? Raúl, ¿te tenemos por aquí? Igual se ha pensado que como estaba abajo no le iba a preguntar y de repente ha bajado. Raúl, ¿puedes hablar o escribir en el chat? Nada. Sara, can you help Raúl? What is the mission?
The question?
What is the mission of the Golgi apparatus, Golgi body?
I don't know. Modifies and package proteins.
¿Ves cómo no era difícil la pregunta? It's going to modify a little bit the proteins in case they need it, but especially it's going to package them. It's going to put them in different vesicles. Why different vesicles? Because the Golgi apparatus is going to build one type of vesicle for those proteins that are needed outside the cell. Secretory vesicles, if the proteins are needed outside, is going to produce a different type of vesicle if the protein is needed in the cell membrane, and a different one if the protein is needed inside. So that's what you've got here. What's the color of the Golgi apparatus in this picture, Sara? What color is it?
Azul, blue.
Así, blue, green, blue, ¿no? Así, una cosa así, ¿no? This one is the Golgi apparatus. This Golgi apparatus is producing three types of vesicles. If the proteins are needed outside, this type of vesicle. If they are needed in the cell membrane, this type that will fuse with the membrane, se funde with the membrane. And the third type, this one, that is when the proteins are needed as digestive enzymes. So this type of vesicle is a lysosome. Okay? Bueno, do you need to know the mission of lysosomes? No, you don't. Just the mission of the nucleus, the ribosomes, rough endoplasmic reticulum, and Golgi body producing the vesicles. ¿Vale? Eso es de las que decíamos de la página 14 que entraban. And the last one, y con esto, bueno, tenemos hasta I25 que me da tiempo a corregiros un ejercicio. And the last one would be this one. There are the centrosomes. Fijaos que vamos aquí y dice, después de coordination, the function of centrosomes. Bueno, pues decíamos que centrosomes are this structure formed by centrioles and the fibers, aster microtubules or aster fibers. These two things, centrioles and aster fibers, formed centrosome, ¿vale? Primeras palabras de esas raras. Centriole, just the yellow one, two centrioles, and the aster fibers form the centrosome. And the mission was forming this structure, the mitotic spindle, that is going to help in the division of the cell. ¿Okay? Bueno, pues yendo a la parte de ejercicios, tenéis que tener todos, tenéis la hoja de ejercicios ahí más o menos a mano, ¿verdad? Decíamos que de esta hoja de ejercicios, the first part is just a review. It's better if you study with your book and the PowerPoint presentation. Mejor, ¿vale? Que aquí viene mucha más información, that you don't need it. And then we've got this part on the chromosomes, and we started, y sé que empezamos porque teníamos, we've got something that is already corrected. Teníamos hasta aquí corregido, ¿verdad? Hasta donde yo tengo en rojo. ¿Cierto? What is a chromosome? Lo habíamos corregido, ¿verdad? ¿Alguien?
Sí, profe.
Eso
Hasta
es.
ahí.
Hasta ahí justo, ¿vale? So, describe, esta era vuestra tarea, ¿verdad? Describe the prokaryotic cell chromosome, que os dije, esto es solo ir arriba, copiar y pegar. ¿Os acordáis? Ay, qué difícil, sin vosotros. Vega, venga, para que me cuentes algo. The difference between chromatin and chromosome, we've already said it in this class, so we're not going to repeat it. Si tenéis duda, pues luego veis el vídeo y copiáis. The difference between this term, chromatin, and this term, chromatid. Vamos a quedarnos con la segunda parte, Vega. What is a chromatid? Any idea?
The chromatid is like
Voy aquí. Aquí.
the one that has the shape of, yes, that one.
Venga, pero dime qué parte es. Chromatids is one half of the chromosome, okay? This half of the chromosome, this part, that is the chromatid. So we can say that is the component or the part of the chromosome that are joined by the centromere. Is the structure that two sister chromatids are joined by the centromere to form a chromosome. That could be the answer. Two sister chromatids are joined by the centromere to form a chromosome. Okay. Bueno, para introducir lo que vamos a ver el próximo día, os dejo una investigación, ¿vale? Que es esto que viene aquí de chromosomal abnormalities. Bueno, the only thing that you've got to do is look carefully these pictures and try to find whether they have 46 chromosomes like humans with no chromosomal problems, or they have more than 46, and if they do have more than 46 or less than 46, where is the problem? In which number? Okay? ¿Entendido, verdad? Count this.
Teacher, yo no lo entiendo.
Bueno, pues eso, count the chromosomes here. Por ejemplo, vamos a hacer este de aquí, ¿vale? Contamos estas. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10.
¿Pero eso dónde está?
En la fotocopia que tienes aquí, donde estaban los ejercicios. Si bajas aquí en contenido, dice cells as a unit of life y dice it includes the activities, que quiere decir que las incluye, ¿vale? Ahí pinchas y te sale esta. Y entonces aquí, en la página de los ejercicios, en la... ya se me ha ido, no. Aquí, en la página 5, empezáis a tener estos dibujos. ¿Qué hay que hacer? Cuentas y dices, este de aquí que estábamos contando, y dices, ¿cuántos tiene? ¿Tiene 46? 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 26, 28, 30, 32, 34. ¿Veis lo que pasa aquí? Instead of 2, it has 3 in chromosome 18. Pues eso es lo que quiero que me digáis. Which one is wrong? ¿Vale? In which one I've got a wrong number. That's what I want you to see. Just pay attention to the pictures and mark the one that has a wrong number. ¿Vale?
And teacher,
Solo eso. No hace falta ni que me busquéis el nombre ni nada. Solo mark the one that is wrong. ¿De acuerdo?
teacher.
Dime.
Y en la que pone X e Y,
Claro, en la que pone X e Y, if it's a girl, it's going to have 2X. If it's a boy, it's going to have like this one. It's a boy because it has an X and a Y. ¿Lo veis? Pues
y
alguno...
si tiene ahí esa mezcla que es
¿Ves?
hermafrodita.
Claro, bueno, no, hermafrodita no, pero ahora te voy a decir. El problema de este estaría aquí, because it has 3 X chromosomes. ¿Vale? Entonces solo tendríais que rodear esto. Venga, que ya os he hecho dos. Este, que es el 18, y este, que es el triple X. ¿Vale? Eso tenéis que hacer, solo rodear lo que veáis que está mal. ¿Vale?
And teacher,
Vale.
el tamaño no importa para que sea una anomalía, ¿no?
El tamaño sí importa para que sea una anomalía, pero en este caso no. Porque en este caso es solo que están arrugaditos. ¿Vale? Este, por ejemplo, es que ha salido arrugadito. ¿Vale? Cuando se estudia sí sería, pero para nuestro ejercicio no. Para nuestro ejercicio solo cuento número, ¿vale? El que veáis que tiene un número equivocado, ese le rodeáis para que mañana lo veamos. ¿Vale? Profe, te tengo claro, si nos vamos todos. Bueno, hasta luego, chicos. Bye, bye.